The introduction. Good morning, everybody. So this is, as Anita mentioned, a three-seminar series. The first two seminars I will present, and then the third seminar will be Dr. John Wilson. Today I want to give an introduction about molecular biology for groundwater scientists, talking about molecular biological tools, the principle of how they work, and what they can do for us to analyze the microbiology in subsurface environments, aquifers. So now we're trying to go to the next slide. Here we go. So just to make sure we're all on the same page, this is a schematic drawing of a subsurface environment, an aquifer. So we have the surface soil, we have a vadose zone, so the non-saturated zone. And then we have the saturated zone here. So this is the zone saturated with groundwater, the water table, and just above the water table where capillary forces pull up groundwater, we have the so-called capillary fringe. Okay, so important here, we have the unsaturated zone, the capillary fringe, the saturated zone, and that's typically confined by bedrock, a confining layer. When we now think about contaminants, we can think about contaminants like petroleum hydrocarbons, and they are released. They are typically lighter than water. They have a lighter density, so they float. So they can form light non-aqueous phase liquids. We think about chlorinated solvents. Chlorinated solvents have a higher density than water. So when they are released, they penetrate the saturated zone, continue with a downward migration, and then pool in areas where they form dense non-aqueous phase liquids. Now these types of contamination can form contaminant plumes. These contaminants are somewhat soluble in water, and you form a plume that can feed from a DNAPL source zone or an LNAPL source zone. Okay, and the plume typically travels with the groundwater flow, in this case from left to right. So now in aquifers, in the saturated zone and the vadose zone, those environments are habitat for a diversity of microorganisms. And just as an example here, I illustrate that there are different types of microbes that make a living in the saturated zone. And those microbes, those are the key targets for bioremediation research, right? Some of these microbes can degrade these contaminants. And that's what I'm trying to explain to you in a little bit more detail. So a good way of approaching this is think like a microbe. And they have one major thing in mind. They need to eat. What can I eat? Where's my food? And in order for them to grow and multiply, they have to do biosynthesis, right? They have to regenerate or generate new macromolecules so that they can divide and grow. And the requirements for growth are, first of all, macronutrients. So those are nutrients that are required in larger amounts, in particular carbon, right? That's a key macronutrient they need in order to grow. But there are others, right? Nitrogen, phosphorus, sulfur, and so on. There's also a series of micronutrients. They're also essential, but they are required in lower amounts. For example, copper, iron, manganese, and so on. So now, when you think about food, what the organism really needs is an electron donor that is being oxidized. So the reduced electron donor is being oxidized by the microbe, leading to the oxidized form of the electron donor. The electrons that are being released are transferred to an electron acceptor. The electron acceptor is reduced, and you end up with a reduced electron acceptor. In this process, the organism can generate energy for biosynthesis function. In addition, the electron donor typically also generates carbon that is needed by the microbes to make new macromolecules and divide. So an example for an electron acceptor could be a chlorinated compound, okay, an oxidized chlorinated compound, and the electrons are transferred to this compound, and the carbon-chlorine bond is broken. The chlorine substituent is replaced with a hydrogen. The chlorine is released as chloride, and that typically leads to detoxification. So now in my next slide, I make this a little bit more easy to understand. So the microbe here's a human who's hungry. So what we need food. The electron donor, here, an example, in hamburger, that's the food we eat. So this is the electron donor, and this is being oxidized. So the carbon in this food is oxidized to CO2. The electron acceptor we are using is oxygen. The oxygen is reduced to water. And what our system, our cells do, they transfer the electrons that are liberated in electron donor oxidation to the electron acceptor. And in this process, we generate energy, and that sustains us. Okay, so the microbe in a subsurface environment has the same requirements. It needs food to get through the day and grow. All right, so that's the strategy microbes pursue, right, in order to make a living. And now, what some of these microbes do, they use contaminants as a food source. And there are different types of microbes that I would like to briefly explain. So we have microbes like this guy here on the top that can use a variety of different electron donors: sugars, organic acids, amino acids, hydrogen. And I should have also mentioned certain contaminants like petroleum hydrocarbons. Those are food sources, electron donors, for this type of organism. But the organism can then also couple this oxidation of these food sources with the reduction of different electron acceptors. The point here is that this type of organism has a versatile lifestyle, meaning it can eat many different things, including the contaminants. In contrast to this type of lifestyle, we also have organisms that are highly specialized. They're picky eaters. So for example, you may have heard about the Helicoccoides is an example for such an organism with a specialized lifestyle. It can only use, now of course I mixed this up here, so my apologies. So the electron donor, they need hydrogen. So they're very specific to use hydrogen as electron donor. And then they can also use only very specific electron acceptors, certain chlorinated compounds. So this needs to be exchanged here. Okay, so they have a highly specialized lifestyle. They can't do anything else. So, just to make this clear, they're very picky eaters. They don't eat everything. They only eat hydrogen and certain chlorinated compounds. And they can't make a living on other food sources. So keep that in mind, a versatile lifestyle and a specialized lifestyle. Both types of organisms with the various lifestyles can degrade contaminants, but this one here can't do anything else. This one here has other options. Okay, so there are different food items on the plate. Okay, so now briefly about terminology. So you probably have heard about these abbreviations, MBTs, molecular biological tools. People also talk about EMD, so stands for Environmental Molecular Diagnostics. So you can use MBTs to do EMD. So what we are mostly interested in in bioremediation is the function of an organism. The function means they are detoxifying the contaminant. Now, the underpinning principles of the phenotype, the function, so the molecular underpinnings of these functional traits, those are the genes and the genomes, the expression of these genes in forms of transcripts, and then the translation of these transcripts into proteins. And the proteins are then responsible for the function, the phenotype. Okay, so the study of proteins is called biochemistry. The study of genes related to function is genetics. And so the area where we investigate genes, genomes, transcripts, and proteins, that's considered molecular biology. So what molecular biological tools do, they interrogate the interplay between genes, transcripts, and proteins. So those are the target molecules that we try to identify, quantify, applying molecular biological tools to environmental samples. So now what are MBTs? They are tools to detect and quantify biomarkers. And the biomarkers, I mentioned it, can be genes, transcripts, or proteins. And the common tool to detect and quantify genes and transcripts is quantitative real-time PCR, or short qPCR. And I'm going to talk a little bit more about this soon. We can also apply metagenomics and metatranscriptomic tools to assess genes and transcripts. And this is something I'll discuss in more detail next week. We can also target proteins using environmental proteomics, another tool I will be discussing next week. So the focus of this talk today will be on PCR approaches. Another tool that is discussed together with molecular biological tools is compound-specific isotope analysis. I will not talk much about this at all, but this is another tool that can be used to demonstrate contaminant degradation. Okay, it measures if bonds are broken. And when bonds are broken, that means the contaminant is destroyed or transformed. And this tool can be used to assess biodegradation extent and also may be able to provide estimates of rates. So coming back to biomarkers, you can distinguish organism versus process-specific biomarkers. Whatever you do with biomarkers, it does require knowledge of the microbiology. So that's a prerequisite. So you have to know the organisms, and you have to know the biomarkers that are on the genome of this organism. And you can use organism-specific biomarkers when you have a firm link between the organism and the process of interest. Remember what I said before between versatile and specific lifestyle. If an organism has a very specific lifestyle, like Dehelicoccoides mccarthyii, it can only do one thing, reductive dechlorination. Measuring the presence of the organism can give you information about the process. If you have an organism with a versatile lifestyle, the presence of this organism does not necessarily mean it actually is involved in the process you're investigating. It can use another food source. Okay, that's an important distinction between Dehelicoccoides mccarthyii and other organisms with versatile lifestyle. You can also think about process-specific biomarkers. So here, a firm link exists between the biomarker, typically a functional gene here, and the process of interest. So as an example, BVCA, VCRA are biomarkers for the reductive declination of vinyl chloride to ethene. Or chloride dismutase is a biomarker for the conversion of chloride to inorganic chloride and oxygen, so perchlorate degradation. So here you have very specific targets for the process. So you can target these genes, and you don't care on what genome they are. So you don't get organism-specific information, but you get process-specific information. So you have to distinguish between organism-specific and process-specific biomarkers. Okay, so now think about the application. So this is just a schematic. These are not real data. But you can go to an environment, take a sample, and ask the question, who is there? You basically characterize the entire microbial community, and then you have a very good overview of who is there. So this can be accomplished by metagenomics or amplicon sequencing. I'm going to talk about these specific tools next week. But you imagine you get a lot of information, and many of the microbes you are detecting have nothing to do with your process of interest. So then, can you be more specific? So this is shown in the middle figure. You only search for those community members that you are interested in. So are the organisms of interest present? And you can do this with organism-specific or process. Actually, in this case, I show here only the organism-specific approach. This can also be done with amplicon sequencing, with metagenomic, or with targeted PCR approaches. You only get information about the three populations that I show here that are implicated in the process of interest. The next approach you can apply, and this is now the process-specific biomarker approach, you don't care what type of organism is present. All you ask is a functional gene that is directly linked with the process of interest present in the environmental sample. So for example, you look for the vinyl chloride to ethene reductive dehalogenase gene VCRA, you detect it, and you don't care what organism harbors this type of functional gene. Okay? And you can tell then if the function is present. This can also be accomplished with metagenomics or with targeted PCR approaches. So why are we doing this? So the results of these MBT approaches can provide information about microbial activities that directly affect contaminant detoxification. You can get information about a site. If biostimulation can work, you may be able to identify bottlenecks, and you can enhance the activity. You may also get information that helps you to decide on the most promising remedy. Maybe you find out that bioremediation is not an appropriate remedy, and you may want to go with another approach. So MBTs can provide information to make this type of decision. Very important, when you do decide on a remedy, you need to convince people that the treatment causes the effect. So you have to establish cause and effect relationships. So the effect means that the contaminants are being detoxified. MBTs can help you to make this linkage between treatment and detoxification. Importantly, you want to predict the trajectory of your plume. So does the treatment provide a remedy for the plume to shrink? And can you make predictions what happens in the next several years? In terms of contaminant longevity, MBTs can help you with making this type of assessment. And of course, you have contaminant measurements, and you may have other geochemical information that points towards detoxification. MBTs can provide an additional line of evidence that you can use to claim that bioremediation at your site is actively working and that contaminants are being destroyed. And I'll give a few examples about biomarkers that we know about and that are of interest for chlorinated solvent degradation. So this is not a complete summary of all the biomarkers that are known. I just want to give you an idea that the science over the last 10 years, 15 years has provided a lot of information about specific biomarkers, organism and process-specific biomarkers that are available to be used and applied for remediation at chlorinated solvent sites. So in the case of chlorinated ethenes, we have a number of organism and process-specific biomarkers for PC, TC to cis-DC dechlorination. So here, organism-specific biomarkers based on 16S rRNA gene targeted analysis. Here I should mention some new findings that are just now being published. We have now also found that organisms that belong to this genus Dehalogenemonas can degrade cis-DCE and vinyl chloride to ethene. So the common belief to date that this final dechlorination step here is limited to dihalocucoides should be revised. There are other organisms in a different genus that have the same capability. So then we also know about process-specific biomarkers, and we have PCA, TCA involved in PC-TC reductive dechlorination. And then of greater importance for monitoring detoxification, BVCA, VCRA as vinyl cis-DC and vinyl chloride reductive dehalogenases. There's another biomarker called TDRA that is involved in trans-DCE to vinyl chloride reductive dechlorination. And then the important ones here for vinyl chloride to ethene transformation, BVCA, VCRA. And then, a new one is CRA. That's a biomarker for this reductive dechlorination step that we found on the Dehalogenemonas genome. Okay, so several biomarkers available for process monitoring. This is a summary, what we know, but again, may not be complete. So we have chlorinated ethanes, chlorinated essenes, chlorinated methanes, chlorinated propanes. We know about the organisms involved, and we can generate organism-specific biomarkers and tools to target those. And then, of course, we can also apply the functional gene biomarker approach to give us additional, more specific information. One thing I should add here is, you may think now, okay, for chlorinated ethenes, the biomarkers have been identified and are known. We have made, as a research community, substantial progress over the last decade to identify these biomarkers. But I don't want to give you the false impression that we know everything. We still are looking for additional biomarkers, new organisms with new biomarkers. What we know today is an incomplete picture. We don't know everything. So there's a possibility that we don't detect any of these biomarkers, but at your site, you may still see ethene formation because we have not identified the key organism or the key biomarker to monitor the process. So it's very important that this research continues And I'm a little worried that too many people believe, oh, everything is done. We can stop funding this research now. I think that's a mistake because the research community should push this further to really make an effort to get the complete picture, the complete set of organisms and biomarkers so that we can improve this approach. And I'll talk a little bit more about this later. Okay, so biology 101. There's a central dogma in biology, and it's the flow of information from DNA to RNA to protein. So DNA can also replicate itself. That's needed when an organism divides and one cell forms two cells. But then in order to access the information on the genome, there's a process called transcription that makes RNA. And the RNA, the mRNA, is then translated to protein. And the protein is responsible for function. So typically catalytic proteins are enzymes, reductive dehalogenases that actually perform reductive dechlorination. And so the DNA can tell us something about who is there. It can tell us about potential activity. Right? So if we detect a certain gene at the DNA level, we know that this gene is there, but we don't know if it's active. Right? Is it transcribed? Is it translated into a functional protein? Well, we could look at RNA. That should give us information about who is active, what genes are transcribed. And this could potentially give us information about the actual activity, not just potential activity. But there are some problems with the RNA approach, and I'll mention it in a second. You could also look at proteins. That means what functions are really expressed. What's the phenotype? And that tells us about the actual activity of the organism of interest. Now, I illustrate this here. This is a cell of D. helicocoides. That's the genome of this organism, this cell. And now there's the 16S rRNA gene that you can target with a biomarker approach, qPCR. Then we also have functional genes, like in this case BVCA, encoding for vinyl chloride to ethene reductive dehalogenase. In the presence of vinyl chloride, the gene is expressed, transcription, the transcripts are going to the ribosomes synthesize the protein. The proteins are then incorporated into the periplasmic space. These are these green balls. And these are the functional reductive dehalogenases that then perform the duty of reductively dehalogenating vinyl chloride to ethene. Okay, so that's the process. The organism sees vinyl chloride. It responds by generating transcripts. The transcripts are going to the ribosomes in blue, making the protein. The protein then is assembled, becomes active, and does its function. The assumption then is, when the vinyl chloride is all gone, it's all dechlorinated to ethene, the transcripts are no longer needed, they disappear, and the proteins disappear. So that then tells us something about the biomarkers. So the DNA is always there, right? But the transcripts and the proteins are only there when the organism is active. Okay, so this is what I mean with, we look at the genome, the genes, we can talk about potential activity, but detecting the transcripts and the proteins would give us information about actual activity, in theory. All right, so now I'm discussing these different biomarkers, the DNA, RNA, and protein level again in more detail. So looking at DNA has a great advantage. DNA isolation is routine. We can do it. Now there are dynamic, and I told you this is only for potential activity. But think about this example of versatile versus specific lifestyle. D. helicocoides, some pseudomonas that can do many things. You see dynamic changes over time. The cells die, they don't grow, or they do grow. And now, just by looking at the DNA in a quantitative fashion, can tell you something about activity. For example, if dihalococoides biomarker numbers increase over time, it means that the organism is active because it can only degrade the contaminant you're interested in. It can't do anything else. So an increase in cell number means it's active. If you apply the same approach to a pseudomonas that can degrade a contaminant, but can also do many other things, you cannot apply or come up to the same conclusion, because the organism may be able to grow on another substrate, not your contaminant of interest. So then you cannot make a link to activity. So targeted quantitative PCR approaches are available that give you absolute quantification, but keep in mind it's a closed approach. What I mean with this, a targeted qPCR approach can only apply to organisms and genes you know about, not to anything new. You can also apply metagenomics, high throughput sequencing approaches. And I'll talk more about this next week. They don't give you quantitative, absolute quantitative information, but relative quantification is possible. And this is an open approach. Maybe not so important for practice, but for research very important, because here we can detect new biomarkers, right? Because we see everything. We don't limit our analysis to genes we already know about. That's why we call it an open approach. Importantly, with DNA, you amplify. You take DNA from the environment, you have very little, you amplify, you make a lot, you can analyze it. So it has a high sensitivity. Many things are also apply to RNA, but there's one important difference. RNA isolation is challenging. RNA is prone to degradation. You lose biomarkers. So this is a major issue looking at this biomolecule. The assumption is that it's highly dynamic and responds to the environment, so the presence of the contaminant, that's an assumption. And it's true for some organisms, but maybe not for all. So linking the presence and abundance of this molecule to activity is maybe challenging. You can then easily quantify it. You get absolute quantification with reverse transcription qPCR, but keep in mind there may be significant biomarker loss. You can also target this molecule with high throughput sequencing. And then what I mentioned, one issue is the interpretation, because we are not sure really if the transcript numbers directly relate to activity. And also this molecule can be amplified, so it can be detected with high sensitivity. Protein. So you can isolate protein from the same sample you isolate DNA. And this is feasible from different matrices. This is accomplished, not a problem. Then again, the assumption is that the protein responds in a dynamic fashion to the environment. The contaminant is present. You find reductive dehalogenases. If the contaminant is not there, reductive dehalogenases are degraded and are not detectable. Talk more about this approach, how this works next time. Just mention that much. The current approaches to measure protein are clearly more challenging than DNA-based methodologies. But this will become easier over time. In the next several years, there will be significant progress. And it will probably be equally feasible to analyze protein. Now, there's one challenge here. In contrast to nucleic acid, RNA, DNA, protein cannot be amplified. So you're always stuck with the amount of protein in your sample. You cannot make more out of it. So that means the sensitivity is a little lower. than what you can accomplish with DNA-based approaches. But again, the technology, the instrumentation has advanced so much that this is not really a major disadvantage, and proteins can be detected in very sensitive fashion in environmental samples. And I demonstrate this next week. So now I want to focus a little bit on PCR. You have heard about PCR, I'm sure. There's regular PCR and there's quantitative real-time PCR. What we typically use is quantitative real-time PCR. Regular PCR essentially is an endpoint approach, and it gives you a yes-no answer. Yes, the target gene is present. No, the target gene is not present. It has a very low dynamic range and is at best semi-quantitative. Sensitivity is okay, but the qPCR approach has higher sensitivity. And important for most of you is probably regular PCR does not generate numbers. Okay? Quantitative PCR, you get real numbers that you can compare over time from the same sampling location. You get absolute quantitative information. It's more sensitive and it has a very high dynamic range. So this is the way to do things. You get quantitative information with a highly sensitive approach. qPCR machines are standard equipment in many laboratories. They have come down in price. There's robust chemistry that can detect amplification. I'll show you this in a second. And the bottom line here is that during the amplification of your target DNA, you generate a fluorescent signal that is directly proportional to the amount of template DNA, target DNA in your sample. What you can do, if you want to learn more about PCR and qPCR, there are very nice YouTube videos. And I just picked two that I liked, where you can see short movies that explain to you how qPCR works. And that's very informative and can help to understand the process in some more detail. Very briefly to quantitative PCR. You have a DNA molecule, you heat it up, you extract the DNA from your environmental sample, you heat it up, and when you heat it up, the double-stranded DNA separates, and what I have in blue here is a single-stranded DNA strand. Then you have a primer that binds to this target template DNA, and you have a probe that binds to the template DNA in proximity to the primer. The probe has a fluorophore and a quenching molecule. In this configuration, there's no fluorescent light emission because the quencher prevents fluorophore from fluorescent light emission. So now you do the amplification. And if the primer binds to your target DNA, the Taq polymerase recognizes these open positions here and extends that primer to replicate the DNA strand. And then it runs into this probe that blocks its path. And so the Taq polymerase destroys this probe, thereby releasing the fluorophore from the quenching molecule. And now you have fluorescent light emission. The point here is the fluorescent light emission is directly proportional to the amount of target DNA in the sample. And that's how you can quantify with qPCR. So this is briefly how this works. You measure fluorescence. The instrument does it for you. These are the cycles in your PCR tube. And at a certain cycle threshold, right? This is a certain number of cycles. The fluorescence passes a certain threshold indicated by this dashed line here. That's called the CT value. And this is what you record. And now you can imagine if you have few template DNA, target DNA, it will take more cycles to generate enough fluorescence to cross that threshold. If you have a lot of template DNA, it will take fewer cycles for the fluorescence to cross that threshold. And that's how you can quantify. Illustrate here in a little bit more detail. I stole this from the web. So you have different amplification curves with different amounts of DNA. Red, high amount, black, low amount. So this is the number of cycles in PCR. It takes a lot more cycles for a low amount of DNA to release enough fluorescence to cross the threshold. With a lot of DNA template, it takes fewer cycles. And you have a standard, prepare standards, right? And you get all these values in between. You can generate a standard curve. And with that standard curve, you can estimate the number of target DNA molecules or genes in your unknown sample. That's how qPCR works. That's a standard procedure. Now, there was in the past some concern about accuracy of qPCR. Right? You can have low accuracy, low precision. The numbers are all over the map. You send the same samples to different laboratories, you get different results. But there are different problems. Ideally, everybody is on target. And so we thought a lot about this, and we had shared projects with different laboratories. And the bottom line is that we have the analytical procedures pretty much under control. So that I would argue the majority of the variability in the analytical results come from sampling and possibly sample handling. So here's a general figure showing how this sampling occurs. You go to a field site, you collect groundwater, or you collect the groundwater, pass it through a Sterivex cartridge to collect the biomass, and ship that cartridge to the analytical laboratory. Storage can influence the outcome, so storage should be minimized. You collect the biomass, if it hasn't been done already in the field, extract the DNA and do the analysis. So most of these laboratory procedures can be controlled, so there's limited variability in the data, if this is done properly. Another issue that was discussed, and that's something you need to think about if you sample an aquifer, groundwater. So this is a sampling well. Here's the screen. You collect with a pump, and you collect groundwater. So recognize that there are cells that are in groundwater, and you suck them up, and you get them into your sample. There are also cells that are attached to solids, the same cells. You will not get those when you sample groundwater. The point is, when you sample groundwater, you do not sample all the organisms that you're interested in from the aquifer. You only sample those that are in the groundwater, not those that are attached to solids. So how does this affect qPCR data interpretation? So the bottom line is, what you may do is you underestimate the true cell abundance in the aquifer. So you get a conservative interpretation of the true abundance because you don't get the attached cells. And so Natalie Capiro at Tufts University looked into this issue in more detail, and that's the paper. And the bottom line is that qPCR may underestimate the true number, but it does not overestimate. So this can have some influence on data interpretation, but it's a conservative interpretation that people will get from the data. And I talk a little bit more about data interpretation next time when I bring this in context with metagenomic data interpretation. So the bottom line is, and this is not a complete picture, it's just some of the literature that comes from my lab that I'm listing here. So we recommend on-site sample collection with Sterivex cartridges. We recommend the TACMAN PCR approach. There was an effort to develop internal amplification control so that laboratories can actually monitor their success with the individual steps involved in the analysis. So that can be done to develop optimized laboratory standard operating procedures. A lot of this was done under CERDA project, and the report is available. And so we do understand a little bit the distribution of target organisms between aqueous and solid phases, and that can also help data interpretation. The bottom line, I think, is that we have good data consistency and accuracy between different analytical laboratories. We achieved that. And so this will further improve through the application of more advanced qPCR technology. So for example, we now have high throughput qPCR instruments where we have these small plates, arrays that basically have more than 3,000 little through holes. This is shown here with a 33 nanoliter volume. And we can run qPCR on these arrays, and we can put up to 4 of these plates in a qPCR machine. So theoretically, we can run 12,000 qPCR reactions in 2 hours. Now this reduces the cost per sample significantly, but because you run so many samples and you have to run so many samples, the overall cost is actually not really reduced unless you really always have that many samples to run, what is unlikely. But what this instrument does for you, it further improves accuracy of the data because a robot does all the volume manipulations, so there's no human error involved. So from the technology side of things, the approach is very advanced, and it's relatively difficult to actually introduce human error into the analysis. Okay, so my point here is that I think the 800-pound gorilla that influences the analysis is groundwater sampling. So it's very important to follow standard operating procedures, such as low-flow sampling techniques. And it's very important that when you do sample a site, a certain well, stick with one method. Don't switch, because if you switch, the data cannot be directly compared. So once you have a standard operating procedures developed, stick with that method, send the samples to the same analytical laboratory, and then you can directly compare the data. So the key question also for you to answer for the field personnel, is the sample that you collect representative of the aquifer formation? So make sure that you don't sample just the well. Make sure that the sample you get is from the screened interval of the well, and you actually collect groundwater from the aquifer, not just from the well. Then we recommend the use of Sterivex cartridges. I mentioned this before, and I tell you why. So what you see here, that's a groundwater sample that's collected, fresh collected groundwater sample. Then this is being shipped on the left side. This is being shipped to the analytical laboratory. By the time we receive it, it looks like this in the middle. This is ion precipitation. Because the groundwater sample, when you sample it, is exposed to some oxygen, ferrous ion will oxidize and form this precipitate. So ferric ion hydroxides. This makes it basically impossible for us to collect the biomass. And then we cannot really extract DNA very well. So that influences our ability to do a good analysis. Now, if you avoid shipping groundwater and collect the groundwater on Sterivex cartridges, so you collect the biomass in the field, we don't have this problem. So keep in mind another thing. If you cannot filter the groundwater on site onto Sterivex cartridges and you decide then to send groundwater to the analytical laboratory, don't expect that they can do any better. They will also have problems filtering the groundwater and collecting the biomass. And I noticed, when I receive samples from field sites, that amendments can make our work analyzing or getting the qPCR data nearly impossible. For example, when we get samples that contain vegetable oil that was injected as an electron donor, it makes DNA, biomass collection, DNA extraction very, very difficult. I had a sample recently. People injected emulsified zero-valent iron, made it very, very difficult to extract DNA. So amendments can impact biomarker analysis. And I think those issues we have to worry about most when it comes to data variability when we apply qPCR approaches. Okay, so we still have improvements for this approach. We need to know more about the microbiology, the ecology. We need more biomarkers. We have standardized procedures, and the goal would be to develop site-specific analysis. We want to understand how these organisms and these genes evolve and disseminate so that we can make better predictions. And we want to link all this MBT information to rates. So all of this can transition bioremediation from a rather empirical approach that I would argue we are employing at the moment to a scientific approach with more predictable outcomes. And this is what I call precision bioremediation. Okay, so we want to transition from an empirical approach to precision bioremediation. And now I'm running out of time, so I'm gonna include more information about what I mean with the term precision bioremediation next week. Where I will also be talking about proteomic tools, metagenomic and metatranscriptomics, and how these tools can ultimately help us to understand the microbial community and better predict detoxification of contaminants, specifically chlorinated effluents. So I'm about a little over my time, so I have to thank many students, colleagues, and collaborators, who generated this information. And then over the years, funding for this work from CERDAP and ESTCP, and then obviously Microbial Insights for hosting me, to giving me the opportunity to talk to you today. And then this may be a time for questions. So thank you very much.
Yes, thank you, Frank, for such an interesting and informative presentation. So we have time maybe for one or two questions. So let's see. The first question is, why is low-flow sampling recommended?
Okay, so we did a study together with Eric Petrovskis as part of an ESTCP project several years ago, where we sampled Eric Petrovskis at Geosyntec at the time, where we sampled wells with different techniques, and we observed that most consistent results were obtained with low flow sampling techniques. When we applied higher flow rates to collect groundwater, there was a lot more variability. And we concluded part of an explanation would be that when you have high flow, you may also get more water from the well, not necessarily from the aquifer formation. So you want to make sure that, that depends on your standard operating procedures. If you use packers to isolate the screen where you want to sample, right, the aquifer, that you get a groundwater sample that represents the aquifer formation. And the experience we made was we got better results using low flow techniques, low flow pumps. And that's published in an ESNT paper in a book chapter that I can make this literature available. So we have very specific SOPs developed that we believe are advantageous to get reliable results for groundwater sampling and subsequent microbial analysis.
Okay, great. So under non-ideal aerobic conditions for dehalogenating bacteria, will their genetic material still be present in measurable quantities?
Okay, so this is a tricky question. So when the aquifer is oxic, and it's oxic for a long time, I would not expect to see high abundance of strictly anoxic anaerobic bacteria such as Dihalococoides. Now recognize, even in an oxic environment, you will always have pockets where you have anoxic conditions. So in those pockets, you will have Dihalococoides and other strictly anaerobic bacteria present, even if the bulk environment is oxic. If you have an anoxic aquifer and then there's intrusion of oxygenated groundwater, so now the bulk groundwater is oxic, but it just transitioned from anoxic to oxic, I would expect you have many Dihalococoides still present. And it will take some time to equilibrate, so for these organisms to disappear, right? So there's a good chance that you will find signals, biomarkers, of Dihalococoides and other strict anaerobic bacteria when the bulk measurements tell you it's an oxic environment. So this is a possibility. So another thing I may add, cells lyse when the conditions are not good, they can't grow. Eventually they die, they lyse, they release the DNA. So the analysis we are talking about here typically only detects intact organisms. So if you have live DNA, that will generally be quickly degraded or eliminated during the sample processing steps. So you don't have to worry too much about naked DNA being a possible factor influencing the qPCR results.
Okay, great. Thank you, Frank. Well, unfortunately, we're out of time today, and we do have more questions that we won't be able to get to. But we'll have Frank review these remaining questions, and we'll email out a document showing both the questions and Frank's responses. So again, we appreciate you joining us today, and thank you again, Frank, for such an excellent presentation, too. So we hope you can join us next Tuesday at 11 a.m. Eastern for part 2 of this series. Thank you.